Well, good afternoon, everyone. Thanks for joining us this almost afternoon. I hope we'll have a good time for this one-woman panel. So, as you have guessed, I will be talking about how climate change is affecting the quality of our seafood by looking at its influence on nutrients as well as contaminants. And before I go into the conversation, I would like to first introduce myself. So I am Amina Shartup, and I am a professor of marine chemistry at Scripps Institution of Oceanography, University of California, San Diego, where I am directing a lab where we look at the global biogeochemical health of our planet through studying contaminants cycling in the oceans. And we have a public health aspect to our research, looking at how humans get exposed to contaminants through seafood consumption. And before you wonder why a marine chemist is talking about health, I have a PhD in marine chemistry and chemical oceanography, but I then worked at the Harvard School of Public Health, where I investigated the impact of environmental change through hydro development on indigenous health. I also worked at the National Science Foundation in their Arctic program, coordinating various agencies that do work in the Arctic and trying to figure out a good way to work in the Arctic with indigenous people and how should scientists behave in the North. So you will see kind of excerpts of some of that thinking through my work, thinking about climate change and indigenous communities in particular through seafood quality. Okay, and this is going to happen a couple times. So I would like to share with you a topic that I think is really important but is not getting as much, discussion as I think it deserves. Many of you have heard that climate change has impact on our crops. It impacts soil organic matter content that impacts how much water can remain in the soils. It also, the CO2 in the atmosphere can impact crop growth. And if it grows faster, it collects less micronutrients in that process. But seafood quality and the impact of climate change on seafood is not something that we typically hear about. And I think that considering an implication for global communities, I think it's really important. And since I have a one-person show here, I would like to kind of engage the audience here and see what you have to say about the topic, but really through mostly raising your hands and answer a few questions. So one of my first questions to you is, how many of you eat seafood? Okay, that's good. How many of you come from a community that is mostly reliant on seafood? Okay, good. And how many of you think about the seafood nutritional quality when you choose which kind to eat? That's good. Do you typically think about how climate change impact that concern that you may all have before you see this? No? Okay, I see some yeses and some nos. Okay, good. Okay. Well, one group that is deeply aware of the impact of climate change on seafood are indigenous community worldwide. These indigenous communities, they have seafood more than just a source of protein. It's a cornerstone of their culture, identity, and livelihood. And so you can see here, these communities are at the forefront of seeing the impact of climate change on their marine ecosystems through having access to seafood that they prefer, but also noticing the change in quality of that seafood. If it was big, now it's smaller, it's harder to find near shore or offshore. And so they definitely have a heightened awareness that stems from this connection to and the importance of seafood, both culturally and for subsistence. You can see also that the communities consume, I don't know if there's a thing here, never mind, consume about 4 times the amount of seafood that the global average. You can see those numbers, about 74 kilograms per capita per year. And they're distributed worldwide around the coastlines. The rest of us, you may have heard that globally about 3 billion people rely on seafood. The map here shows the per capita seafood consumption across the planet. You can see that there's a wide variation in that consumption of seafood, where we see a lot of dark blue, so more than 50 kilograms per year, per annum per capita, in Southeast Asia, Japan, Portugal, and some of the northern Scandinavian countries. And we also see that there are countries where seafood consumption is not as widespread. That's often due to the just geographic location where they're far away from the oceans, or culturally, this is just not part of what they eat. But as you look at that map, you can see that we mostly have blue. So a lot of the world population is dependent on seafood, but also that the impact of seafood quality will be disproportionately hit some of the communities that are bluer. There's also, as I said earlier, a variation within each countries where some coastal and indigenous communities may be more impacted by changes in seafood quality. So seafood, of course, plays a critical role as a source of protein, but it also is an essential source of nutrients and micronutrients to billions of people worldwide. So I put a list of the nutrients that come from seafood, and I highlighted in orange and underlined those that are almost exclusively available through seafood consumption. So you cannot easily get that from something else. And so that would be some of the fatty acids that you may have heard, like omega-3s, DHA, EPA, etc., or iodine. Obviously, you're ready for the bad news. That's why I'm here. Seafood is also a primary source of a wide range of toxicants, and that includes, I kind of separated them in several categories here. Toxic metals, you probably heard of mercury, which is the #1 reason for seafood consumption advisories on the planet. We have persistent organic pollutants, and as well as a range of emerging contaminants such as microplastic and pharmaceuticals. You will notice that on the list of my toxic metals, I have selenium in parentheses. That's because selenium is actually a micronutrient at low concentration, but it can become contaminants at higher concentrations. Although this is not typically what we see in marine environments, we don't have excess selenium. Okay, so another important aspect of the puzzle is that with few exceptions, seafood is a net positive in terms of health, despite the presence of contaminants. This is because it provides the essential nutrients that I mentioned that are essential for brain development, immune function, which as infectious diseases rate is increasing, having immunodepression is a concern, so making sure that people are getting the zinc and the iron that seafood is rich with is something that public health experts are concerned about. And as the diet is shifting globally and people are moving to urban settings and they're having a more modern diet, we are seeing large shifts in access to micronutrients, and globally we see a decline in micronutrient access and an increase in deficiency. So this is a very small map, but it doesn't matter because red means bad, and this is mostly red and orange. So most of the global population is deficient in iodine, and I think The Economist had an article in the US that came out recently that as people are getting into those artisanal salts and so on, they're no longer getting their iodine, and now the US population is mostly depleted in iodine. So finding ways to address those growing nutrient deficiencies, something that many people in the public health world are concerned about, and one of the major way that people have proposed to address that in the study by Hicks et al. Is to consume fish. And so here they calculated what the consumption of seafood collected within 100 kilometers of near shore. And I see some people don't have headphones. You probably would want to have headphones if you want to hear me speak. There should be some in the back there. You can get access to most of those nutrients fishing sustainably within 100 kilometers of their coast. So that addresses a lot of micronutrient deficiencies. However, one aspect that they did not address in the study is, just like I said, as much as we are getting all these micronutrients from the seafood, we are also collecting those toxicants, and particular neurotoxicants of interest to me is methylated mercury. And so this is some work that got published about a month ago by Ming Li, looking at how much of that methyl mercury humans are taking out of the ocean through fish that they then go home and eat. And so, and you can see that there are definitely some hot spots around the regions where we fish a lot. So it's about areas where we go out to get some tunas and other large predatory fish. And as we are collecting those micronutrients, we're also collecting all these contaminants. So the question that is unclear and we are actively working on, and I will not have an answer for you today because I'm just here to share some of the problems. I may have some answers for you today, is that climate change is going to impact how these both nutrients and those contaminants are distributed globally and the way we are collecting them. And I'll get into details as to how exactly this is going to happen in a few minutes. I'm going to give you a few examples of the interplay between those things, and I will also share some of the investigative tools and techniques that we use to understand and answer this question. And yes, I will not necessarily give you some solutions, but I'm hoping that you will be feeling better equipped to go and advocate for us to get more funding to explore that thing that is really important for everyone who consumes seafood. So one example here is some work by Colombo et al., where they investigated the availability of DHA, which is one of those fatty acids, omega-3s, that is essential. We cannot de novo produce DHA, which means from scratch. We can only get it from our diet. Most things cannot produce DHA. It's almost exclusively produced by algae in the oceans, which is beg the questions, how did we evolve to get to this point? And normally animals would still over time build up a little bit of that DHA from consuming certain plants that have another form of omega-3s, and that they can slowly convert it. But unfortunately, we're now feeding a lot of our animals grain, so they're not being able to accumulate the DHA within poultry or pigs or beef that we consume. The supplements are not a solution because often the supplements are coming from the ocean anyway. So if we see declines from ocean omega-3s, we're going to see a problem concentrating them in form of supplements or even in farmed animals, which are actually fed wild seafood collected animals, powdered and fed to the farmed animals. So the farming is also not a solution to that. So this is a real problem that people are grappling to address. So phytoplankton, algae produce that DHA and then they pass it up the food chain. So then we eat those animals that are higher up the food chain, and that's how we get our DHA. However, the processes within the marine system that generate that DHA are highly sensitive to temperature because the phytoplankton produces in the cell membrane, and that cell membrane changes depending on the temperature of the water, and so that is going to have implications for the amount that they accumulate eventually. So in this study, they use a relatively simple approach where they looked at how much DHA we get out of our seafood and they looked at a linear relationship between the base of the food web and DHA production and temperature. They made some assumptions, like we are going to continue to fish from the same location, the fish is not going to move, the phytoplankton structure is not going to change, and they make some prediction based on temperature of seawater, how much DHA is going to end up. And again, you don't need to look at it. It's red. When it's red, it's bad. And you can see that everywhere, regardless of the scenario, we see that we are more on the yellow and orange. So we are seeing a decline in DHA at a global scale in our seafood. So, okay, so at the same time as we see the declines in just one example of a micronutrient, but I'll show you another example later where they are indeed declining, and probably worse than what this prediction is, because I'll tell you a little bit more about other issues that we need to consider, but in this particular study, they just look at the linear relationship with temperature, but those systems are a lot more complex than that. So at the same time as we're seeing declines, we're emitting and releasing and concentrating a lot of the contaminants in the environment. And I'm giving you an example here of a study done by Charlotte Wagner, where she looked at PCB's distribution in the oceans. And as you can see, we've been emitting those starting sometimes in the 1930s. They peaked as many things in the 70s. They kind of been declining, but really they've been mostly entering the deep sea where they've been accumulating, and they're substantially above, well, background, because background is 0 for PCB because it's human-made. But the deep sea is important in terms of accumulating those contaminants, but also because a lot of the fish that we like to consume eat from the deep sea, and so we may see an increase or a kind of like a stabilization of the concentration. And that's just one example. Another example is our emissions of mercury, and this is the one I'll focus on here because this is kind of the core of my expertise. And mercury has some of the most sophisticated models for contaminants biogeochemistry that we are looking to adapt to some of the other elements that we are interested in studying. And so that's why I think it's a good example. In addition, mercury is actually one of the best marker for seafood consumption in humans. So if I take a human and I ask them, do you eat fish? Well, I don't, because I can just measure their mercury content and tell approximately how much seafood they consume. There are some studies looking at the best predictor, human biomarker of seafood consumption, and mercury is by far the best correlated with seafood consumption. So contaminants released like a mercury follow pretty well the emissions and greenhouse gases. That's because mercury is emitted primarily through coal burning. And so as you burn coal and emit CO2 through that, you also emit a bunch of mercury. And the trends are quite similar, and you can see that we are definitely peaking up there starting in the 1850s. And so today, the estimate is about 90% of the mercury cycling on the planet is been put there by humans. It follows relatively complex biogeochemical cycle. So once it enters the ocean or the ecosystems, climate-driven changes are going to impact its distribution and how it behaves in the environment. And so from temperature to fire to permafrost thawing to sea ice to changes in temperature. And I'll tell you a bit about how this is happening, but bottom line is that we are trying to model a lot of these processes happening at the same time to see what is the ultimate outcome in the seafood and what will be the future trends. And we can't really answer those questions without understanding how all of these changes impacting the mercury cycling. Okay, so a little bit about the mercury cycle. As I said, mercury is emitted primarily through a range of anthropogenic activities. For a very long time, it was emitted by coal burning. But recently, there has been competition for artisanal gold mining that is now estimated to be the #1 source of mercury to the environment. The question there is difficult to really put a number on it because a lot of the mercury used in gold mining is illegal. So it's very difficult to calculate exactly how much is coming through gold mining versus how much is emitted from coal burning. But that mercury gets emitted. Mercury has a unique characteristic that it's a volatile, well, it's a liquid metal, so that's why people were really excited to play around with it. But a big fraction of it is volatile. And so it gets emitted as this elemental form, and it has a really long residence time in the atmosphere, which means it spends a lot of time just floating around. And so the sources are kind of, could be, in the middle of the Pacific, but your mercury can land right here in Azerbaijan, and we'll have to deal with the consequences of it. Once it's emitted, there's a bunch of atmospheric chemistry that happens to it, and a fraction of it can be either reduced or oxidized, and it then deposits when it turns into a stickier form of mercury. So any weather pattern will impact the mercury deposition. So as climate is changing and the weather pattern, the precipitation pattern are changing, it's going to impact where the mercury deposits. So this is something we need to think about as we study it. Okay. Once it deposits, and if it reaches those aquatic system, unfortunately for us, the microbes start taking over and messing with it, and so that causes an additional layer of complexity for us to understand, because the form of mercury that actually ends up in the fish is not that mercury that we emit. It's a methylated form of mercury, which is microbially produced naturally in the environment. So the changes in those microbial community structure are going to impact how it enters the food web and how much of it ultimately ends up in fish. And of course, as you all know, changes in seawater temperature, nutrients availability has huge impact on microbial communities. So this is something that we need to worry about as mercury scientists. Okay. It doesn't stop there, unfortunately for us, is that once it enters the food web, it biomagnifies, which means that processes that drive the prey-predator interactions, any changes in food web structure is going to impact how much mercury ends up at the top of the food chain. So if a predator runs out of a certain prey and switches to another prey because we either overfished it or that prey moved because it no longer likes the water temperature that it's in, that predator will have a different concentration to mercury. And then finally, humans are finicky. We have preferences that are driven by fads, Instagram, TikTok, all of that good stuff, and people start eating certain things that they normally weren't eating. So depending on how the wind change, humans will consume different types of seafood. For a long time, we didn't consume things like bluefin tuna and things like that. And then there was develop a taste for it, and now everybody's eating sushi and everybody's eating tuna. And at least in the US, it's the #1 source of mercury to our diet. So these things can also impact exposure to mercury. Okay, so as I kind of briefly talked about, and I'll go really quickly, these are the various forms of mercury that we're dealing with. These are the ones you're familiar with on the left, which are the metallic, silver, liquid one, and the volatile forms are the one we emit. The microbial communities then take that up and they convert it to these organic forms, which are actually the ones that most of us consume. So when people talk about mercury in fish, they're not talking about the ones on the left, it's only the ones on the right. And then we have those processes of transformation and biomagnification. So at each step of the food web, so most of the uptake of methylmercury occurs in the phytoplankton, which multiplies the concentration within the cell by maybe 10,000 to 100,000 times, and then it starts increasing up the food chain and reaches millions of times in higher trophic level predators. That's why people tell you don't eat sharks. I'll give you a quick 2 examples of how climate change directly impacts that and the type of work we've done to address this. One example was some work we did for UNEP AMAP program, where they asked us if we could predict what is the mercury concentration in the Arctic going to be like under a range of climate scenarios as well as emission scenarios. And this is particularly important for the indigenous communities in the Arctic because they wanted to know, okay, what are we dealing with here? And so what we did is that we built a pretty simple model looking at mercury cycling in the Arctic Ocean. And then we borrow from the climate people and the CO2 emissions people some of their scenarios, because like I said, the mercury emissions and the CO2 emissions are linked, so we can kind of use the same prediction. So if we move away from coal burning, we know we're going to reduce, and so we can use their policies to apply to our emission scenarios. And so we have some scenarios that we run, and then we also look at changes in climate-driven variables such as sea ice area declines in the Arctic, or river discharge changes, or net primary production, which we also borrow from the IPCC predictions. And we look at how the combination of these things impacts mercury concentration in the Arctic Ocean. And generally what we find is that, kind of not, surprising news, is that if we don't do something, it's not good. We'll see really high increases of mercury concentration in the Arctic Ocean. If we do anything to curb the emissions of CO2, aka Hg, mercury, we will see declines in the Arctic Ocean concentrations of mercury. The sooner we act, the larger an impact is going to be. The longer we wait, the smaller the impact is going to be, or the more aggressive we are going to have to be to curb the concentration. Again, very similar to the conversation that we are having for CO2. And in parallel, so if something is on the right, it's bad. If something is on the left, it's good. That's pretty much where you want to be. For the climate, they have an impact on the mercury concentrations. The sea ice reduction reduces the mercury levels in the ocean. Some of the other ones increase. So the climate impacts in the Arctic are kind of in both directions. Overall, at least in the Arctic, it seems to be helping a little bit in terms of mercury concentration at the surface. But at depth, we're going to see higher concentrations of mercury because all that phytoplankton that is now blooming in the Arctic, sucking up mercury at the surface and actually dragging it down to the deep ocean. So we may see increase in deep ocean mercury levels and decline in the surface. Again, bad, good sign. Okay, so on the biomagnification side, which is kind of the other side that I wanted to show you what things look like, we are looking at how the methylmercury that is being produced is being potentially affected by climate change. And so there things can get really complex because you have all these interactions between different prey and predators. And luckily in the Gulf of Maine where we're doing the study, they've been looking at stomach contents for about 50 years. So we have an idea of who is eating whom and how that thing has changed, how the predator-prey interaction have changed, and how the diet of the animals has changed. And I'm sorry if I look like this because they're like making a lot of noise next door and I keep getting distracted by it. So we created that virtual aquarium because a lot of these animals can live 20, 30 years. So it's really difficult by just measuring the condition on the ground to be able to tell what they're exposed to over that time. So we created this virtual aquarium where we can modify the seawater temperature of our aquarium. We can modify which prey is added or removed and how ultimately the seafood methylmercury concentration changes. And so this is kind of a summary of the things we're considering. We're considering the bioenergetics of the animals. So, how much do they eat and how much do they lose, how quite quickly do they grow, and then we also considering the food web interactions, who eats whom and how much of the different species, and then we ultimately look at the concentration of different animals as they grow and age and eat each other. And so things that are interesting that we need to consider is the hunting strategies of the different animals. So depending on their hunting strategy, they will take on more or less mercury. For example, a swordfish, they can eat a slightly larger prey because they can potentially knock it around. And I know if you saw the story where somebody got hurt by a swordfish, got killed recently. Other animals are restricted by the size of their mouth, so the biggest animal they can eat is the size of their mouth. So if they have a small mouth, they can't take on a lot of mercury. But if you're a scavenger, you can eat something big that died, like a shark, and you will have a high. So these are all the things that we need to consider when we're building our predator-prey interactions and thinking about mercury transfer through the food webs. So how can temperature impact things? So this is an example of cod. So this is a base scenario where we measure, we model how mercury accumulates under basic 2000 conditions where they're eating their base diet, and then we can modify to a different diet and see how methylmercury concentration change. In this case, we fed them a 1970s diet in the model, and we can see that the mercury concentration go up. We can increase the temperature of our aquarium seawater and see what happens to our mercury, and you can see that it goes up. And then ultimately, if you look at a fish that we are all interested in, which is Atlantic bluefin tuna in this case, we can just look at projections of if you increase the temperature of our ecosystem by 1 degree Celsius, how much of the increase in methylmercury is going to be driven by the temperature alone, assuming we curb emission and they stay flat. And we show that we can see a substantial increase, all other things being equal, and keeping emissions flat. Temperature alone can increase the methylmercury concentration. And this is driven by animal bioenergetics, that as you warm up the water, their energetic demands increases at the bottom of the food chain, and that propagates up the food chain, and that results in accumulation of more methylmercury up the food chain. So what does it mean going back to a micronutrient issue? Well, it means that this is a base scenario where we are looking at, so the study by Milling again, where they're looking at selenium to mercury ratio in the seafood that we fish. And then they're also looking at omega-3 to methylmercury ratio of the same seafood that we fish out. And as you can see, again, red is not good. So in this case, red means you're getting a lot more contaminant relative to the good stuff that you want. And you see that those equatorial region, we get a lot more contaminants per unit of the good stuff. And so as I'm talking about, we're seeing those decline, they didn't do any climate scenarios, but as I'm talking about, we see those decline in those omega-3s, and we're seeing an increase in those contaminants. We should really be concerned about the changes in our seafood quality as we go forward. Selenium, I wanted to mention here that if you look at the selenium to mercury ratio, you can see that, it's not as bad. We see a bunch of blue everywhere, especially in the northern countries over there. But we did a study fairly recently looking at about 40 years worth of selenium data, working with the Swedish Museum. And so we looked at the selenium trends there in the Baltic Sea herring, which is extremely prized. And selenium is a micronutrient, like I said, and it's also an antagonist to mercury. A lot of people say that they think that it counteracts the neurotoxicity of mercury. So you want to have as much selenium per unit of mercury as you can to kind of counteract those impacts. And we see that over the past 40 years, driven by changes in agricultural practices, primarily in Finland, when they used to spray selenium everywhere, and our regulation of the acid rain, so sulfur emissions being controlled means that selenium that is also emitted from coal burning at the same time as the sulfur is trapped by the same traps that are controlling the sulfur. And so we are seeing the combination of this global reduction in selenium emissions, and the local practices are changing, and so we see those declining trends. So of course the question at the end of the day is, can we put these all together? So in the study that I showed you about the omega-3s, they kind of did those linear relationship, but we know that not all the algae, not all the phytoplankton produces. We know that those communities are going to change. We know that some fish are going to move around. We know from the work that we are doing in mercury that changes in the way the food web processes are going to impact how much accumulates. The same processes are also going to impact how selenium and omega-3s are accumulating in the seafood. So we can see forward that those things are going to change. So we recently received support to do this work through the Scripps Center for Oceans and Human Health that is supported by the National Institute of Health in the U.S. as well as the National Science Foundation. I'm leading the project on the climate change impact on seafood quality. So we're really excited to starting doing the hard work of putting it all together into very similar models where we can start teasing out what direction is the planet going to go in terms of seafood quality. And on this, I would like to thank you, and I'll take any questions that you may have. I put some picture of some of my fantastic students and postdocs and all the people who are doing the hard work. Of course, the funding, the money people are important. Thank you.
Presentation. A couple of quick questions on methyl mercury. So is the question, first one, is it accumulated only in the oceans, or like is it same in the freshwater fish as well?
Actually, that's a fantastic question. I mentioned earlier that mercury is causing a lot of the seafood advisories. It's actually primarily in freshwater systems. So a lot of areas that have historically gold mining or any factories that like felting industry, well, you know that the mercury poisoning disease is named after Minamata disease. And so this was a factory that was spewing right into, in this case, it was a bay. But a lot of those same factories that are placed all over, lakes and rivers can result in long-term contamination of mercury. And like I said earlier, I did some work in the north in Canada looking at the impact of hydroelectric development on indigenous health. And in that particular case, it was reservoir creation for hydroelectric development because they flood a bunch of soil. And like I said, that mercury goes up, deposits all over the place. Once you flood and you create a reservoir, essentially a lake, the microbes that I was complaining about earlier, they take up that mercury and make methyl mercury that then ends up in the fish in the reservoir. And there's been cases of displaced community in Canada due to reservoir creation and methyl mercury pollution. Yeah.
Thank you. Sorry. My second question is more a bigger picture question. Obviously, climate change is the reason here. So do you have any other potential solutions that we can do actually to reduce this methyl mercury?
Again, really good question. So I usually have some slides of things that we are trying to do. So one thing that we are clear about, and I think this is very similar to the CO2 community, carbon community, is that we need to reduce emissions, control emissions. And this is work because we now have the Minamata Convention to control and help countries reduce the mercury usage and reduce the emissions. Now, the problem is that we've been doing it for hundreds of years, so we have a lot of legacy mercury that is cycling. That mercury is going to take thousands of years before it's removed through geological processes, so it's not going to be anytime soon. So the question then becomes, what do we do? Well, so there are things we can do. So one thing we don't tell people ever is to not eat seafood. So that's, as I said, I hope it was pretty clear that eating seafood for a lot of the communities is extremely important. And there's no replacement for the nutrients. So especially in areas like the Arctic and a lot of, poorer communities where whenever you tell them not to eat seafood, the alternative is usually worse in terms of nutrition than the seafood, even if it has PCBs and mercury and PFASs in there. But one thing that we are working on a project with UCLA collaborators that we were trying to figure out if we kind of leverage the human microbiome, because some people are able to break down some of the methylated mercury and we're not quite sure why. And so we have recently engineered a microbe that can in vivo demethylate mercury following seafood consumption. We've tested it in mice. So these mice are microbiome free, but we recently applied for funding from NIH to see if we can try to test our microbes with a human microbiome within those mice models. We've had some really good results in terms of reduction in methylmercury in the body parts and organs of the mice, but also lower fetal transfer because we were looking at how much ends up in the fetus in mice that have the microbe and the ones that don't. So that's one of the kind of solutions because we were like, what can we do? But we are thinking, at least I'm thinking in the long term, we can think about ways for us to potentially do a better job at extracting the little bit of nutrition that we have through microbiome intervention from other things. I know that people, for the DHA issue, people are working on creating microbes, microbial produced DHA, because they're realizing that we will run out of seafood-based, marine-based DHA. So there's some engineering interventions there too. Oh, 1 second.
Thank you for a fascinating talk. I just wanted to ask you, for parts of the world that don't have very good baseline data on mercury levels, what are recommended indicator or sentinel species?
Yeah, so, geez, my voice. Okay, so this is a huge problem, which is one reason I've been kind of running around and trying to say hi to every pavilion from any country where we are lacking data. The species is a concern. So we've been working, so my collaborator at Scripps, Anela Choi, we actually work on a paper right now looking for new forms of biomonitoring species that we can find all over the world. So we are looking at potentially using some really low energy deep sea fish as a bioindicator. And the reason we're looking at that is because you saw that the bioenergetics are quite important and the food web interaction are quite important. And this deep sea fish is extremely low energy. So we don't have this like increase in methylmercury as they grow because all of a sudden and the other thing is that they eat very little and their stomachs are like refrigerators. So when you get their stomach, you can identify every species of fish that they've eaten. So we're just finished writing the paper. We're looking to submit it. We're looking to potentially propose that as a good biomonitor because it's found globally and it get caught in the same fisheries that catch the tunas that you see are the dominant form of methylmercury removal process from the ocean. There's no easy answer in terms of some of the other stuff. I think just having one of the things I'm working with one of my students right now, we're going to go test it out in Nigeria in February, is portable instrumentation. So we're kind of working on the instrument development part to see if we can easily measure the concentrations in areas that we think a lot of gold mining may be happening or potentially a lot of contamination, but they don't have necessarily the resources to bring in some ultra high purity argon to run mass specs. So we're going to test that out this February, and we're hoping that our little portable systems are going to work. So we're trying very different things and see what's going to stick.
Hi, as someone from an equatorial fishing-based economy, I'm curious to see what are some of the research or pilot programs that could be done in order to really expand upon the scope, and what are some of the pathways for individualized medicine approaches? Like, I know that the mice study is there as well, but like, what other components are there?
Yeah, so one thing that we've done, not in this particular case, but working in the Arctic and also with my colleague who is from Hawaii, is to just go to markets and ask people to give us a little bit of what they eat at any given time so we can kind of get an idea of the typical exposure that people have from just what they collect at the market. So I have a philosophy about generally working in some communities like that is that I never go to ask. I wait for them to come and ask, just because, we're scientists, we come, we just bug everybody, and everybody's like busy trying to live their lives. So I don't want to bother anyone. So usually the way things happen is like when we did the work in Labrador, they were well informed as a community because historically this has happened to some of their cousins nearby. And so they came to us and they said, hey, they're setting this up. We think it's going to have an impact. And the industry is saying they did a risk assessment and there's going to be zero impact. We don't believe them. Can you come and do the work? And so we were like, okay, cool. Like, we'll come and we'll do the work. And obviously they were right. The other guys were making stuff up. They had no data on the ecosystem. So when they're making predictions about what's going to happen, it was based on dilution is the solution. They're like, yeah, there's going to be production of methyl mercury in the reservoir, but it's going to go and mix in with which just happened that system was highly stratified. So whatever was being produced just kind of like sat on top where the phytoplankton lived. And so it was ridiculous. But so this happens. But I think that a lot of these things is I go around and I try to tell as many people that this is something that we should look into. And I'm hoping that some people will hear and come back and be like, hey, we want to do work here. Yeah. We have lots of time for questions, so don't hesitate.
Many thanks, Amina. Indeed, it was really an outstanding presentation, an outstanding work behind it. When I think about your results, and I'm trying to understand how we can apply this to management of the ocean. And of course, there is a lot of work in the Food and Agricultural Organization about fisheries management. There is definitely some need to support artisanal fisheries. Mm-hmm. But we know what is going to happen in the coastal waters, including also medications and things like this. So also there is some consumption not only of fish, but some other animals. Mm-hmm. So, is it possible to basically, start generalizing your conclusions? So you can come up with some recommendations to those who manage fisheries and aquaculture in terms of what is going to happen, not in relation to climate change, but overall with the total trends that we are experiencing. Needed.
Yes, really good question. So yes, so one of the component of that project that I'm leading that we just got funding for is to do exactly that. Is the idea is that we're building those models and we can start thinking of predicting about, well, this type of fish are going to see this decline in this nutrient, but an increase in this contaminant. Should we eat something else as an alternative? And so we're trying to have some sort of advising tool. So we as part of the SCOOSH or whatever, the Center for Oceans and Human Health, we have a community engagement component project. So it's one of the 4 projects, and their job is to try to take our science and translate it to actionable things, working with local fishermen and seafood consumers to think about what are the useful ways that we can present that information that can be actionable on their end with the fisheries. So we're working with NOAA Fisheries Management, that they can extract information that is useful for them. Because some of the difficulty here is always translation, right? So we have the science, it's great, but how do you translate this into something that is not harmful? And that is something that a colleague and myself wrote a piece about recently, is how do we make sure that in our discipline of geohealth, we are not accidentally harming some of these communities by providing half thought out advice. So we're really cautious, which is one reason I come here and I say, look, at the end of the day, right now, as things are, seafood is a great source of nutrients and people should stick with it, because we haven't figured out a good way to pass on that information without people getting scared and so on. So I think there's a huge role for social scientists and humanities here to help us produce useful, helpful advice. Yeah.
Thank you for your presentation. Did you see any ecological aspects that can be can be used as an indicate between species, between the relation between predator and prey? Because we do monitoring the environment. We monitor the local fishermen. We know where they are fishing, and you see any biological indicator by a species number are decreasing or increasing population of determinate species to be an indicator of the ratio of mercury in the environment.
Well, so the paper that I mentioned here, we tried to do a little bit of that. It wasn't very deep, but I think there's definitely room for improvement. This is, so I had multiple fish actually, so I'm just showing you dogfish, but we had cod, and we saw that when you switch their diet, so what happened in the 70s is that we had a herring crash, and because of overfishing in that area, and so the main predator from this particular prey chose different alternatives. And depending on which one they chose to eat, the methylmercury either went up or down. And so we answered in a way a long-standing question that we had in the communities, that why do you have the same ecosystem, same deposition patterns, same trophic position predator, completely opposite trends in mercury concentration, and this is driven by their prey choices. So we could do that. We could say, like, if we have good fisheries data, which we had here, this is what they're eating today, but that stock is declining, and this is their alternative prey. What are going to be the methylmercury levels in that fish? We can have a pretty good idea of what that is, as long as we have some bioenergetic information about that animal. Or at least we can say it's going to go up and down. But some of these systems are so complex, you have so many other forcings, that it's difficult to say it with a high level of certainty without a long-term data to support it. So I would say, again, it's always about baseline monitoring, surveying, having enough sample per age group, size, etc. And of course, finding the good biomonitor for these things is something that we are still working on, as I answered, I think it was your question here. So I think we can do things. We just need to figure out how to do it. And the mercury community is relatively healthy as far as contaminants are concerned. But as you saw, the cycle is really complex. So everybody's working on a little bit of it. And so it's difficult. Like, we are very few that work across all of these things, are thinking about, from the policy aspect all the way to the seafood. So it's pretty much like my group and maybe one other group that is looking into these things. So if we are more of us, maybe. Yeah.
Thank you for your brilliant presentation.
Thank you.
I was just wondering, I've been working in places like Greenland and the Faroe Islands where there's, a culture of higher consumption of apex predators, and something that I've encountered in the Faroe Islands was that there's, they'll take into consideration whether, women of reproductive age will not eat whales, for example. But these sorts of things are not necessarily taken into consideration in Greenland. And someone was telling me that there are other aspects of the diet that can sort of counteract some of these. One of the things that I was told was that canned tomatoes, for example, because in the communities in Greenland you won't get a lot of fresh vegetables, if any, has some aspect that, and I'm not sure, I'd like to clarify if that is the case.
Yes, thank you, Larissa. It is the selenium. So people are saying that, so mercury toxicity, the way it works is that methylmercury enters crosses the blood-brain barrier through mimicry. It looks like an amino acid. It tricks the body. It lets enter into the brain. Once it gets there, it has a really strong affinity for selenium, like super strong. Selenols and thiols, so sulfur or selenium protein. These enzymes, their role in the brain is to deal with the reactive oxygen species. So the brain is a super oxygen demanding organ. And so there's a lot of reactive oxygen species that are being formed. You need some cleaning protein in there doing their work. Methylmercury messes with those. It binds them, it prevents them from doing their work. You get a lot of reactive oxygen species that causes oxidative stress, and that's where the neurotoxicity comes from. So people are thinking if you consume enough selenium, you will counteract some of these impacts in two ways. One, it will bind to the methylmercury or the mercury in your digestive system before it gets there, and so it will just sit there and do nothing. Or two, you will just have a high supply of selenium, so you'll be able to produce more selenium enzymes that will help counter. It's highly controversial in terms of science. There are people who are saying that's not a thing. There are others that are saying that's a thing. The experiments definitely need to be, a lot of them are really old, so it's unclear if it's still the case if we do it again. So, but there hasn't been too much work, but the little bit that I've seen more recently, they're like, we're not sure. So I think that this is an important question that should be addressed. And I think that people need to look at other ways that we can counteract the impacts of methylmercury, not just through selenium. But I bet you this is what people are talking about. And one of the evidence for that is that several marine mammals that accumulate a lot of mercury, they do have a demethylating, detoxifying mechanism. So they form these mercury selenium compounds, and we see nanoparticulate mercury selenium in their liver and kidneys and so on. So they are actively doing that. We know it from just looking at their at their livers, but also through we use mercury isotopes. And you can see that there's a very clear signature on the mercury isotopes that is a signature of demethylating in vivo. So, and that's why my collaborator ran with that. Is there a way we can reduce methylmercury exposure through that action? And hopefully that could be an alternative to switching from traditional foods, which again, like I said, are culturally and health-wise really important. And we got 2 minutes left, so we could do one last quick question, or we can say bye-bye. Oh, one last quick question.
Since I'm from the organization called Maldives Space Research Organization, we're trying to do a lot of EO remote sensing in order to kind of at scale monitor these bioaccumulative. Are there any biomarkers that could be kind of identified at that scale?
Yes. So one thing we could do is, so one of the things we were looking into is for areas where we don't have a lot of data, if we have an idea of the amount of dissolved organic carbon, because like I said, that mercury is super sticky and it likes to bind to thiols, in particular, and organic matter has a bunch of those. And organic matter has a color. So we use those color-based satellite to kind of calculate the dissolved organic matter content and then use that as a proxy for how much mercury may enter. So that's how sometimes we do the work in the Arctic to calculate the export from the Siberian rivers. So that's one way to do it. It's highly uncertain, but it's better than nothing in some regions. So yeah. Okay. Thank you.